My volume seems to be very loud. Okay. All right. Let me know if you guys can hear me.
I can hear you.
Great. Before we begin, are there any questions? All right. I'm not hearing any questions, so let's begin. I mean, make my screen available to you. Oh, that's not right. Hopefully I didn't log out. Nope, I'm having trouble getting the Zoom screen for some reason. There we go. All right, you should be able to see my screen now. Is this the last slide we talked about? Can somebody confirm that?
Yes, that's accurate.
Okay, great. All right, so we talked about phospholipids and that they are the main molecule in the cell membrane, and they're actually what makes the cell membrane. The phospholipid has a phosphate part, and that'll be on the outside of the membrane, as well as the phosphate head on the inside of the membrane. And then the fatty acid chains of the phospholipid will be inside the membrane, away from the liquid that'll be outside the cell, and the liquid, which will be water, inside the cell. So the next lipid we need to talk about are steroids and sterols. If you remember, I think I showed it at the very beginning here when we were talking about lipids. There are simple lipids, complex lipids, and steroids. A steroid is a lipid, but it does not have any monomers. So the one molecule makes the steroid. Cholesterol is a steroid-like molecule. It does have an OH group attached to it. And if there's an OH group attached to it, we call it a sterol. So cholesterol is a sterol. Sterols are an important part of the animal plasma membrane. As I mentioned before, they intercalate inside the phospholipid bilayer, and they give the cell membrane strength. So steroids are a large molecule. They have four interconnected rings and then a few carbons attached to it. And cholesterol is the most well-known molecule in this group. Any question about steroids and sterols? All right, here's a question for you. Cholesterol is found only in what foods or in what cells? You do not find cholesterol in certain foods. You do find it in other foods. So what foods do you find it in? You guys should know this because cholesterol, well, it's not uh, the health hazard that we thought it was, but it still is not something you want a lot of in your diet. In what food do you get cholesterol in?
Is it fatty meats?
In animal products. And yes, you get more in fatty meats because cholesterol is a lipid, so it'll be dissolved in the fat. And obviously, you'll get it in any meat, even lean meat, because it is in the cell membrane of animal tissue. But you do get a lot more of it in fatty meat because it will dissolve in the fat. Any question about any of that? So if you want a cholesterol-free diet, and actually when I was younger, like in the 1980s and maybe the 70s, they were promoting a cholesterol-free diet. And all you had to do was eat a vegetarian diet and it would be cholesterol-free. Because plants, as well as algae, do not have cholesterol. Only animals have cholesterol. Any question about any of that? All right, let's move on to the third group now that we're talking about. We talked about carbohydrates, we talked about lipids, so let's talk about proteins. Proteins are an essential part of cells, their cell structure, and proteins are very important in the functioning of cells and organisms. Proteins do many different things. There are structural proteins, and you can find these proteins in bone, muscle, and hair. There are also proteins that are an enzyme, meaning they make up the enzyme, and they speed up chemical reactions for the cell or the organism. There are proteins that transport molecules across the cell membrane. We haven't talked about it yet, but the cell membrane regulates what comes into and what leaves the cell. And if the cell membrane doesn't allow the molecule to cross it, which, except for small nonpolar molecules or lipids, the molecule cannot cross the cell membrane. And so if we want the molecule to come into the cell, like glucose, you have to have a transport protein. Or if you want the molecule to leave the cell, let's say some nitrogenous waste product, you need to have a transport protein. And so the cell puts a lot of transport proteins in the cell membrane to pump the molecule either into the cell or out of the cell. Any question about any of that? And obviously they're very important for an organism or for a cell to function. And then there are regulatory proteins. These regulate body functions, such as hormones. In children, they produce growth hormones, and these growth hormones help the child grow. Any question about them? And then there are proteins responsible for movement, both when people walk or just my moving my hand. That's because of the protein in our muscles, as well as for cells to move like a bacteria. If it's flapping its flagella, well, that flagella is a protein that allows the cell to move. And that's true for sperm cells and humans, they have a flagella. And then also for the cilia, like on our throat. All this movement is because of the protein molecule responsible for the movement. Any question about any of that? And then there are proteins that are responsible for some bacterial toxins. Most exotoxins are a protein, and they are toxic. I can't think of one off the top of my head. I can think of several proteins, but I don't know if they're, excuse me, I can think of several toxins, but I don't know off the top of my head if they are proteins or not. I'm going to guess, but I'm not positive that the toxins in some E. coli are proteins, as well as the toxin in Corynebacterium diphtherium, but I'm not positive. I just don't know the toxic substances. What's the shot that we get for like if you step on a nail or something like that?
Tetanus.
Tetanus. I think tetanus is a protein, that toxin. And then there are antibodies, which are also proteins, and these proteins help defend an organism against infection. So humans make antibodies, and these proteins or antibodies help defend the body against an infection by a foreign agent, such as bacteria. Anyways, you can see that proteins play a large number of different functions in an organism, and proteins are more diverse than any other of the four biological molecules. Proteins do consist of a monomer, the subunit that we call the monomer of a protein, we call them amino acids. And the reason why proteins differ more than all the other biological molecules is because proteins have the most monomers. Humans have 20 different amino acids, which get together to make the protein. When we're talking about carbohydrates, there's a handful, maybe a dozen of different sugars, and that's true for most lipids too, although they're closer to a dozen that make up the different fats. And proteins, at least in humans, have 20 different amino acids. Different species tend to have different amino acids, but most of them, if they choose another amino acid than what humans have, they throw out one amino acid and then add another amino acid, so that the organism, which is a bacteria that has the most amino acids, and you don't need to know this, I'm just telling you, most animals, and most, not animals, but most organisms have 20 amino acids, and they just throw out one if they put in another amino acid. But there is one bacteria that actually has 23 different amino acids, and you don't need to know that, so don't write it down. Don't, I won't quiz you on that. An amino acid is shown here. It's a generalized amino acid. You'll note all amino acids have a central carbon and a hydrogen attached to that central carbon. And then on the other side of that central carbon, we have an amino group, that's NH2. And on the other side, we have a carboxyl group, CO with a double O, and then OH, which is most often called COOH. And that's a carboxyl group. And then we have a fourth group attached to that carbon, and that we just abbreviate R. R can stand for anything, and it's actually R which makes the amino acid, meaning all the amino acids have all the other parts, and they just differ in what R group they have. So for tyrosine, it has this R group. The simplest R group is glycine, which simply has an H for where the R should be. Let me see if I can blow this up. So glycine has an H in that R group. Some of the R groups are nonpolar, like alanine and valine and leucine and isoleucine. These are nonpolar R groups, as well as glycine. But note that an amino acid is always polar. The amino group and the carboxyl group are polar. And some amino acids have a nonpolar group, which will be in the R region of the amino acid. Come on, mouse. There we go. You don't need to know the different amino acids, so don't Don't try to remember them. I won't quiz you on that. And there's another nonpolar R group. This R group is both nonpolar. This region of the molecule is nonpolar, but this region of the molecule is polar. And then this R group right here, this region here is polar and that region is polar, but this carbon there, that carbon there, this carbon here and that carbon is nonpolar. Any question about any of that? So the point is that proteins are both polar and they have regions which are nonpolar. Not all amino acids are nonpolar, but there will be nonpolar amino acids in a protein because the protein will have 20 different amino acids. And with a protein, when it folds up, if the protein is in water, the nonpolar regions of the protein will fold up in that protein so that the nonpolar regions are inside the protein. And then the polar regions are outside the protein. And then the protein will have the nonpolar regions acting with the water, like in the cytoplasm, which if you don't know, cytoplasm is mostly water. So any protein which is in the cytoplasm will fold and the nonpolar regions of the protein will be tucked inside the protein. The polar regions will be on the outside. If, on the other hand, the protein is in a lipid, such as the cell membrane, the protein will fold up so that the polar regions of the protein are tucked on the inside, and then the nonpolar regions of the protein are on the outside, and the nonpolar regions of the protein will interact with the lipid bilayer, which is nonpolar. Any question about any of that? All right, amino acids are linked together by a peptide bond. What happens is we get two amino acids which come together, and the OH leaves the carboxyl group, come on, mouse, of one amino acid, in this case glycine, and an H leaves the other amino acid, in this case alanine. The H leaves from the amino group to form water, and then that nitrogen will bind directly to that carbon, shown here. And this is called a peptide bond, and it's made in dehydration synthesis when two amino acids link together. A peptide bond is very uncommon both in living organisms as well as in the universe. That normally nitrogen does not often bind to carbon. Okay, but it does happen in amino acids and proteins, and that's why it's called the peptide bond. Any question about any of that? All right, to make the protein, you just hook all of the different amino acids together, so they'll be linked together with the peptide bond. And then you have the monomers forming a polymer, and the polypeptide chain will be the growing protein. And when the protein has attached the last amino acid, we then call it a protein. So when the protein is being made, we call it a growing protein. We don't call it a protein. You can call it a polypeptide because that's what it is. But after the last amino acid is added, we then call it a protein. Any question about any of that? All right. Proteins have different levels of protein structure, and there are actually 4 different levels to the protein structure. There's the primary level, the secondary level, the tertiary level, and the quaternary level. let's watch a little video talking about these different levels.
When the sawdust settles and the engine roars, the thing you care about is a job well done.
Sorry. It comes from YouTube, so you
In
had to
this
watch a
video,
little
we're
bit
gonna
of the
talk about
commercial.
the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid.
All right, let's move on because I've already covered that. I want to talk about the different level of protein structure.
We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you can see that it's stabilized by hydrogen bonds. The NH group of one amino acid.
All right, let me state that that's the main bond in the secondary structure. However, there are other bonds, which this video does not talk about, that form in the secondary structure, and I'll talk about those.
Interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we can see some areas, it's just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, I didn't notice that earlier, but he actually mentioned a third shape of the secondary structure. There's the alpha structure and there's the beta sheet. And he pointed to here and said, there's a third structure. And I'm going to call that a random coil. I'll show you what a random coil is in just a minute. Any questions about this video? Like it says, it's very good, but it does have one error in it. And it's not really an error because the hydrogen bond is the main shape, is the main bond giving rise to the shape of the secondary structure. But there are other bonds which give rise to the secondary structure. And I shouldn't say bonds, I should say there are other interactions between amino acids that give rise to the secondary structure of the protein. So as the video showed you, the primary structure is just the sequence of amino acids in a polypeptide chain. So when the protein is finished being made, all it'll have is the primary structure, the sequence of the different amino acids. And that would be this amino acid followed by that amino acid, followed by that amino acid. Any question about the primary structure? The different proteins will have a different primary structure. And of course, they have different sequence of amino acids. The secondary structure is the localized interaction between different amino acids, meaning nearby amino acids start interacting with each other in the protein. That gives rise to the secondary structure. The interactions that can happen between the nearby amino acids are, number one, the hydrogen bond. So this amino acid is polar, that amino acid is polar, and they form a hydrogen bond between them, which helps stabilize the secondary structure. It also helps form the secondary structure when that hydrogen bond forms. And there's the beta pleat showing you a hydrogen bond. And this is the alpha helix having a hydrogen bond. But there are other interactions that can happen in the secondary structure that happen between nearby amino acids. Like these two are not attracted to each other. They're actually repulsed from each other. And that's why they're away from each other. This repulsion could be that this is a non-polar amino acid, and that's a polar amino acid. They don't like being together, so they'll be repulsed from each other. They can also be repulsed if this is a positive amino acid, and that's a positive amino acid. We will have a positive repulsion from them both having a positive charge. Any question about any of that? And that's true if this is a negative amino acid and that's a negative amino acid. They'll be repulsed from each other. So those are some of the different interactions between neighboring ones, but there's still another one. And that is if you have two amino acids, let's go ahead and put this one there because they are close together and they're being attracted to each other, but it doesn't have hydrogen bonds. Both of these could be a non-polar attraction, meaning non-polar molecules like to be around non-polar molecules. And they have an attraction to each other that we call van der Waals forces. We're not going to talk much about that. And I'll bet you didn't have much of it in Biology 160 either. But when you have your oil and your vinegar together and you shake them up, everyone knows the oil will separate from the vinegar, which is mostly water, and the water will be on the bottom, the oil will be on the top. And you all know that the water is a liquid because of hydrogen bonds holding the water molecules together, forming the liquid. And if you didn't have those hydrogen bonds, you wouldn't have a liquid, you'd have gaseous water. Any question about that? Well, let's get back to the nonpolar molecule interaction. Why do the oil molecules come together and then stick to each other to form a liquid? Because of oil, the molecules were not to stick together in some way. They would be gaseous oil, which you wouldn't see that separating in your salad dressing because the oil would all evaporate if it was gas. And what happens is the oil molecules have van der Waals forces, and then they hold together, forming a liquid. Any question about any of that? Anyways, the point is that these amino acids, if they're nonpolar, they will be attracted to each other by a very weak interaction that we call van der Waals forces. I think that's all of it. Let me see if there's any more that I forgot. Hydrogen bonds, which happen between two polar molecules, and one has to have a negative end, like a partial negative end, like partial negative oxygen, and the other has to have a partial positive end, like a partial hydrogen. Hydrophobic interactions I just talked about. That's the two nonpolar being together. And then ionic bonds. Actually, I haven't talked about one, and that's the ionic bond. If this is a positive charged amino acid, well, that would be a bad way because those are the same color. Let's go this one here. If this is a positive charged amino acid and this green one is a negative charged amino acid, they will form an ionic bond between each other. And that will be an attractive force. And then I mentioned, like, if this is a positive amino acid and that's a positive amino acid, they'll be repulsed from each other. That's not an ionic bond, it's actually an ionic repulsion. Anyways, those are all the different type of interactions you can have in a secondary structure. And all of these interactions only occur between nearby amino acids. So in the secondary structure, it's only localized interactions between neighboring or nearby amino acids. And that gives rise to the secondary structure. And you were already told the alpha helix, the beta pleated sheet, and then the third structure, which wasn't directly mentioned in the video, is just a random coil. So I'm going to draw something. And this random coil, let's go ahead and bring this over this way too. This random coil can have interactions right here, and then right here. And there might be repulsion in these regions or no interaction. Okay? Any question about any of that? But the point is this amino acid is right there, and it can have interactions right there and right there. And we call this a random coil. Now, once the secondary structure occurs, then the tertiary structure can form. The tertiary structure is when amino acids in any region of the protein chain can interact with another amino acid in the protein chain, and they can be at any position in the protein chain. For example, this amino acid is at the end of the protein chain, and this one is near the beginning, and they're being attracted to each other, and that's why we call it the tertiary structure, because these amino acids are not nearby, but they are interacting to each other. The tertiary structure can happen when the amino acids start interacting, and they can be all of the interactions we discussed in the secondary structure, meaning they can be hydrogen bonds, they can be ionic bonds, they can be van der Waals forces, bringing the amino acids together, or they can be forces pushing the amino acids away from each other, and that can be a nonpolar region with a polar amino acid, or they can be an ionic repulsion where the amino acids both have a positive charge or both have a negative charge. Any question about any of that? So all of the forces of attraction you can find in the secondary structure, you can also find in the tertiary structure. The difference is in the tertiary structure, these amino acids can interact with another amino acid anywhere along the protein chain. And that will cause the supercoiling of the protein into the three-dimensional structure of the protein. Any question about any of that? All right, there is one additional force of attraction that we only find in the tertiary structure, and that is the disulfide bonds. Cysteine has a sulfhydryl group attached to it, and you can have a cysteine here and a cysteine there, and the sulfhydryl groups can lose the hydrogen. Let me draw that out if I can get it. Oh, there it is. So we can have our protein like that, and the protein like this. And right here we have cysteine. I'm going to put the cysteine right there. Let's see if I can copy that. Paste it. See if I can bring it over. Yep. Well, maybe I can move it this way. Yep, I can. Excuse me. And I'm going to try and draw the sulfhydryl group because it's going to be, yeah, that's not very good. Yep. So it has a sulfhydryl group on it. And this one does too. Come on. Bring that over there. Yep. What happened to my hydrogen there? I don't know. Try that again. All right, these hydrogens can come off. And then the sulfur can hydrogen bond with another sulfur in cysteine. I'm just going to draw that in. And this we call the sulfur actually, I don't want to save that. Let's go over here. This we call the disulfide bond. Let's see if I can show that here. So here we have the cysteine here, and the hydrogen came off, and it'll just go into solution. And then the hydrogen came off here, and then they form the covalent bond between sulfur. This is a very strong bond. And for those of you who've had your hair permed, you know that this bond is very strong because when they perm your hair, what they're doing is they're making a disulfide bond in your hair. And this bond can last about a month, even when you shower your hair. Whereas in the when you curl your hair, my mother used to put her hair in a curling iron, so she'd put him in the curl, and I don't remember what you call it, but there would be a little thing she wrapped the hair around, and then she put a bobby pin to hold it in place, and then she put the cap on, and then she turned on the machine to put steam in her hair, and then she exposed it, I don't know for how long, 10, 15 minutes, something like that. And then she would stop it and then pull out the curls, and her hair would have this wave in it. And that was forming only a hydrogen bond. And of course, she lived in Western Oregon. And so when she did that, and then she walked out into the misting Western Oregon rain or the misting Oregon weather, the curl would just sag. And that's because hydrogen bonds are very weak. And even a little bit of mist in the air would break that hydrogen bond. Whereas when you get your hair perm, you can take a shower and your hair will still be perm because it'll have a disulfide bridge in the hair. Any question about any of that? Anyways, those are the different attractions and repulsions that give rise to a tertiary structure. Remember, the tertiary structure are the same attractions and repulsions in the secondary structure, with the exception of the disulfide bond between cysteine, which only happen in the tertiary structure. But all the other are the same as the secondary structure. The difference is in the secondary structure, the amino acids have these attraction and repulsions between neighboring amino acids. And in the tertiary structure, these attractions and repulsions can be between any amino acids in the protein chain. Any question about any of that? All right. For many proteins, all they have is up to the tertiary structure. They don't have the quaternary structure. And that will be every enzyme I know of. And that's because every enzyme I know has only one protein subunit making up the protein. However, there are some proteins that have more than one protein subunit. And that would be something like an antibody or hemoglobin. And both of these proteins have 4 protein subunits that need to come together. So there's one here. Come on, mouse. There's one there, there's one there, there's one there, and there's the 4th one there. And these will have amino acids in the different protein subunits interacting with each other. Like these amino acids are attracted to each other. These will be repulsed from each other. Once again, these are attracted to these, etc. Anyways, the point is the coming together of the different protein subunits to make the final protein is the quaternary structure of the protein. And all of the interactions we saw in the tertiary structure happen in the quaternary structure. And I just told you hemoglobin and antibodies have 4 protein subunits. You could call them 4 polypeptides, whatever, that need to come together to make the finalized protein, which we call the antibody or the hemoglobin. And that's the quaternary structure. Any question about any of that? Remember, only some proteins have a quaternary structure, but all proteins have a primary, secondary, and tertiary structure. All right, talking a little bit more about proteins. proteins can have a overall shape. And if the overall shape of the protein is roughly compact and roughly spherical, we call that a globular protein. Hemoglobin, and almost every enzyme you can think of, is a globular protein. But we do have other proteins which are thread-like or fibrous-like proteins, and these tend to be connective tissue proteins, like collagen is a fibrous protein. With proteins, we can have the protein connected to another part of a different molecule, and we call these conjugated proteins, combinations of amino acids or proteins with other organic or inorganic components. These conjugated proteins are named by their non-protein component. Like glycoproteins are a glycogen connected to a protein. Lipoprotein is a protein connected to a lipid. And you can find both glycoproteins and lipoproteins in the blood, as well as the cell membrane. Any questions about any of that? If not, we can talk about protein denaturation, and that is a protein in the correct environment will have a three-dimensional structure where the protein, amino acid protein chain will be folded in on itself to give rise to a three-dimensional structure, which if the protein only has one subunit, we'll call that the tertiary level. But if the protein has more than one subunit, we'll call that the quaternary level of the protein. You can have the protein lose its shape and not being in its correct folding pattern, and that we call denaturation of the protein. When the denaturation is severe enough, the protein will no longer function. In the denaturation, you can break hydrogen bonds, you can break ionic bonds, you can break, obviously, van der Waals forces easily, and that can give the protein, cause the protein to lose its three-dimensional shape and be denatured. However, you can also cause the protein to become denatured if you form bonds that should not be there. Like our cells, if you were to take your tissue, obviously you have to cut it out of yourself, but you take your blood, for example, out of yourself, and then put it in the refrigerator or the freezer, it would form hydrogen bonds that should not be there. And that could cause protein denaturation also. So when the protein becomes denatured severely enough, the protein will no longer function, and that we call a denatured protein. Denaturation can happen when the protein is in the wrong environment, such as the wrong temperature, the wrong pH, the wrong salt concentration. An example of this is when you crack an egg and you pour the egg white onto the pan, and then you heat up the pan, you're denaturing the egg whites of the protein. And so the egg whites of the egg will be mostly clearish. It'll be a liquid. And then when you cook it, the egg whites will turn white as well as will become a solid. And what you're doing is you're denaturing the three-dimensional folding pattern of the protein, causing the egg whites to denature. And that's the wrong temperature. If you don't know, you can also do this with changing the pH. I don't know how many of you have ever done this, but has anyone ever pickled eggs, we're talking about like a raw egg. If you put an egg white or egg whites in vinegar or something that's acidic or extremely basic, you put it in the wrong pH and you can denature the egg whites. And then it'll stop being kind of clearish and will turn whitish. It will become more solid, but I don't think it'll become a total solid. And that can happen also if you put the egg white on a whole bunch of salt. That would be something you guys could actually experiment with. You can get a little bit of your egg white when you're making an egg and put in a whole bunch of salt, and you'll see the egg whites denature and turn white. I don't think it becomes a total solid, but it does become thicker. Any question about any of that? So a protein has to be at its correct environment in order to have its correct three-dimensional folding pattern. In an incorrect environment with the incorrect temperature, the incorrect pH, or the incorrect salt concentration, the protein will start to lose its shape and will start to become denatured. All right. Any questions about proteins?
I have a question about another one about egg white. Is something similar happening when you make like meringue, when you beat the egg white until it gets white and fluffy? Is that also denaturation?
I think so, but I don't know that one off the top of my head. But
Okay.
you do bait the egg whites and it changes color. It's obviously white.
Mhm.
It doesn't permanently become a beat egg white, meaning if you get it enough time, it will tend to separate and become regular egg white, or not a regular egg, yeah, egg white, meaning like egg whites out of the egg. But I think not all of it will become the reverse of denaturing, whatever that's called. I don't know. And so it's not a permanent reaction when you beat the eggs. Is that clear?
Yes.
All right,
Okay.
great.
Yeah.
But I think it is denaturation, but like I said, I'm not positive because I don't know what's going on when you're beating egg whites.
Okay. I'm gonna Google it because now I'm curious.
Okay, go ahead and
Thank
give
you.
us the answer. All right, let's move on to nucleic acids. There are two nucleic acids, DNA and RNA, and you can call them this. You can also call DNA deoxyribonucleic acid and RNA ribonucleic acid. Each of these nucleic acids consists of monomers that we call nucleotides. And nucleotides consist of a 5-carbon pentose sugar, shown here in blue, and then a phosphate group, shown in pink right here, and then a nitrogen-containing base, which there are 4 of them, and one of them is shown here, and the other one is shown, well, 2 of them are shown here. There's one there and one there. Any question about any of that? So nucleic acids are made up of nucleotides. The nucleotide is the monomer. The nucleic acid is the polymer. And there's only two polymers you need to know of, DNA and RNA. Although it is true there are different polymers of RNA, but they have the same chemical structure and makeup. We'll talk about different RNA in a minute. Any question about any of this? All right. DNA and RNA are made up of nucleotides, and they are each made up of 4 different nucleotides, which I didn't mention that with protein. Remember, I made the statement that human proteins are made up of 20 different amino acids. And I did say carbohydrates, there's from a handful to a dozen different simple sugars that make up carbohydrates. Well, with nucleic acids, there are only 4 different nucleotides in each nucleic acid, meaning each DNA and RNA have only 4 nucleotides. The nitrogen-containing base, often just called the base, can be either a purine or a pyrimidine. For DNA, the purines are adenine or A and guanine or G, and you can learn either name, either adenine or A. The pyrimidines in DNA are cytosine and thymine. And I'm going to give you a little trick to help you remember that a pyrimidine, what's a pyrimidine and what's a purine? In DNA, the pyrimidines always have a Y in the name. Like pyrimidine has a Y in the name, cytosine has a Y in the name, thymine has a Y in the name. The purines do not have a Y in the name. Adenine and guanine do not have Ys in the name. Any question about any of that? Now for RNA, they have 3 of the same bases, meaning RNA has adenine or A, guanine or G, and cytosine or C. They do not, however, have thymine or T. So thymine is not in RNA. Instead of thymine, we replace it with uracil. And uracil replaces thymine, so uracil is a pyrimidine. Any question about any of that? And like I said, the easiest way to remember the pyrimidines from the purines are, say with DNA, if the nucleotide has a Y in the name, then it is a pyrimidine. And for RNA, you can use that terminology. It's just that it doesn't have thymine in it. So that's removed because there's no thymine in RNA. It's replaced with uracil, and uracil is a pyrimidine. Any question about any of that? All right. In DNA, the bases bind to each other via hydrogen bond when they are in different strands. So I guess what we should talk about first, let me see if I got it here. There we go. One strand of DNA is held together by covalent chemical bonds, holding the DNA together. So one strand of DNA is held together by covalent chemical bonds. And it's only held together by the sugar phosphates. Let me blow this up. So there's the, it's hard to say because I don't see what it is, but the sugar phosphate. The nucleotide bases are on the steps out from that single strand. And the nucleotide bases hydrogen bond together to make the step of the DNA double helix. So the guanine and the cytosine and the adenine and the thymine do not hold the single strand of DNA together. What they do is they hold 2 strands of DNA together by hydrogen bonds. hydrogen bonds are much weaker than a covalent bond. So a single strand of DNA, at least on the surface of the Earth, is always a single strand of DNA. And the hydrogen bonds at room temperature can break, like this one in the GC can break, but the others hold, and these hold, and those hold. And then this can reform, and then this bond up here can break. And that we call DNA breathing, and that is the DNA will have its hydrogen bonds breaking and then reforming. Any question about that? The single strand, I should say the double strand of DNA is held together, at least at body temperature and room temperature, because there are so many different hydrogen bonds that even if this one breaks, there are other hydrogen bonds around it holding the DNA together. And then this one can reform and that one will break. And maybe this one up here is also broken. But like I said, the DNA will be held together at body temperature because most of the hydrogen bonds will be forming hydrogen bonds, and only a few of them will be broken at any one point in time. And so the DNA will be held together by hydrogen bonds, the two strands of DNA. If we were to heat that DNA up, like put it at 70 degrees or higher up to about boiling, you can get the DNA to be single-stranded, meaning you get it to 70 degrees or higher up to about boiling, all of the hydrogen bonds will break, and then the DNA will be totally single-stranded. On the planet Earth, that's how you would get, but if you were to go to the sun, you could then break off the single strand of DNA, break off the different nucleotides in the single strand of DNA. The sugar in DNA is deoxyribose, which gives the DNA its name, deoxyribonucleic acid. The DNA is double-stranded in DNA, and it is the sugar phosphate backbone which holds together a single strand of DNA. The bases on the single strand are protruding outwards, and two strands of DNA will have two bases, one in each strand, hydrogen bond to make the step. And DNA actually forms a twisted ladder much like this. All right, RNA has the sugar ribose. RNA, at least in our body, is always single-stranded. But that's not quite true too. Our RNA is always single-stranded. And I'm wording it like that because later on in the term we'll talk about viruses. Viruses can have RNA, at least RNA viruses do. And RNA is a little bit different, and we'll talk about that when we talk about viruses. Any question about any of that? So RNA is single-stranded in our cells, and once again, the RNA is held together by the sugar phosphate backbone. So that if we were to say this is a strand of RNA, it will be single, there won't be two strands there. And it's the sugar phosphate backbone of the single strand which holds the RNA together. RNA does not have hydrogen bonds with each other. RNA does have three major kinds of RNA: transfer RNA or tRNA, messenger RNA or messenger RNA or mRNA, ribosomal RNA or rRNA. And each of these RNAs are important in protein translation. And we'll talk about that in a little bit. So that's the importance of the different types of RNA. Here's a question for you. Actually, there's two questions for you. The first question is, let's see if I get it right. How does a cell know to make tRNA? Well, no, let me back up a minute. What's the difference between tRNA, messenger RNA, and rRNA? What is the difference between those forms of RNA? All right. If you guys aren't going to guess, we'll just leave it. But this actually is a fairly important question.
Doesn't
Say again?
doesn't tRNA it transcribes like the sequence, and then the mRNA has to do with amino acids?
The tRNA brings in the correct amino acid on the growing polypeptide chain, and that's because the tRNA does read the message in the messenger RNA, which determines what the protein will be made, meaning what amino acid needs to be put in will be brought in with tRNA. So tRNA is important in protein translation, if that's what you were trying to say. And what
Yes,
about
thank you.
rRNA and messenger RNA? Are they important in protein translation? Come on, I just told you. All of these three RNAs are involved in protein translation. The tRNA brings in the correct amino acid and actually does the reading of the messenger RNA. The messenger RNA codes for the sequence of nucleotides, or excuse me, the messenger RNA codes for the sequence of not nucleotides, amino acids in the protein that is being translated. And then the ribosome RNA is important in making the ribosome. The rRNA gets together with the ribosomal protein to make the ribosome. And the ribosome is what's allowing protein translation to occur, meaning protein translation only occurs at a ribosome. And you need all three of these types of messenger RNA to come together to translate the protein. The rRNA gets together with the R or ribosomal protein to make the ribosome, and then it moves down the messenger RNA, allowing the tRNA to come in and put in the correct nucleotide. But what's the difference between the tRNA, messenger RNA, and rRNA? Do they have a different sugar? Does one of them not have ribose as a sugar?
I would say no.
Yeah, that's a no. And that is they all have the same sugar. I told you RNA is made up of ribose for its sugar. And they all have the same 4 nucleotides as well, because RNA only has 4 nucleotides, which will be a nucleotide of adenine, a nucleotide of guanine, a nucleotide of cytosine, and then a nucleotide of uracil. So it has the same 4 bases. So what's the difference? Anyone have a guess? All right, the only difference between tRNA, messenger RNA, and rRNA is the sequence of nucleotides. If the sequence is in the sequence for a tRNA, then we call it a tRNA. If the sequence is in the rRNA, then we call it rRNA. And both tRNA and rRNA only have a handful of genes which make the tRNA or the rRNA. And the only difference between tRNA, rRNA, and messenger RNA, once again, is the sequence of nucleotides. But there are many different messenger RNAs or mRNA, because each protein that differs will need a different mRNA. But once again, the only difference will be the sequence of nucleotides in the RNA. All right. The DNA, when it's copied, all of the DNA will be replicated. So when DNA is replicated, all of the DNA will be replicated, both the genetic material and the non-genetic material. And we'll talk a little bit more about that later. In RNA, is all of the genes, is all of the DNA, I should say, is all of the DNA transcribed to make the tRNA, messenger RNA, and rRNA? Anyone have a question there? So I'm trying to ask you what DNA is transcribed to make the different forms of RNA. Is it all of the DNA which is transcribed, or only some of the DNA?
I would say it's only some.
Yeah, it's only some. The genes for tRNA, the genes for messenger RNA, and the genes for rRNA will be transcribed to make RNA. All of the genetic material, which is not a gene, will not be transcribed. And for bacteria, most of the DNA is genetic material. But in eukaryotes, most of the genetic information, or most of the genetic DNA, sequence of DNA, does not code for a gene. Any question about any of that? So in DNA of eukaryotes, most of the DNA is not genes. In bacteria, most of the DNA is genes. And here's your last question. We'll see if anyone answers this one. And that is, how does the gene that's being transcribed know to make tRNA, messenger RNA, or rRNA? So when the gene is being transcribed, how does the gene know to make tRNA, messenger RNA, or rRNA?
Is it based off of the sequence?
Yeah, it's based off the sequence in the gene. If the gene codes for a tRNA, then its sequence will code for tRNA, and tRNA will be transcribed. However, if the gene codes for ribosomal RNA, then rRNA will be transcribed. And if the gene codes for a protein, then messenger RNA will be transcribed. So the polymerase doesn't need to know what type of tRNA to make, because it's just reading the DNA and then adding the RNA, and it'll make either tRNA, messenger RNA, or rRNA, depending on what the gene codes for. Most genes code for messenger RNA. Only a few genes code for tRNA, and only a few genes code for rRNA. And then this is just something you can know for trivia, and that is most of the RNA in your cell is actually ribosomal RNA, even though most of the genes code for messenger RNA. Is that clear? All right. And like I said, I won't quiz you on that last part, but I may quiz you on how does the gene know which RNA to make? And it's just whatever the gene codes for. And then what's the difference between the RNA molecules? It's only the sequence of the RNA. There's no other change. All right, let's talk about ATP, adenosine triphosphate. It is the primary energy source for all for most cellular activities. So if the cell needs energy, it typically gets its energy from ATP. And that's why we call it the energy source or the energy currency of cells. ATP has ribose, that sugar right here. It has adenine in it right here. That's a nucleotide base. And it also has 3 phosphate groups. In ATP, the 3rd phosphate group has a squiggly line because that's a high energy bond. And the 2nd phosphate group also is a high energy bond. If the cell needs energy, it'll break off this 3rd phosphate group to get the energy the cell needs the energy for. And if the cell needs further energy, it can actually break off the 2nd phosphate group to get even more energy. And like I said, these are high energy bonds. ATP is made in dehydration synthesis. What you do is you get ADP, adenosine diphosphate, add on a phosphate, it's an inorganic phosphate, and then add in energy. And then you can get the reaction going this way, forming water and ATP, or adenosine triphosphate. And then when the cell needs energy, it'll add in water and go this way, breaking off the 3rd phosphate group, which will release a lot of energy, and the cell can use some of that energy for its energy needs. Of course, some of that energy will be lost as heat, and that's just the 2nd law of thermodynamics. All right, the last slide in this lesson is what molecules have we discussed have hydrogen bonds that give the molecule important properties? So what molecules have we discussed in this lesson that have hydrogen bonds that give the molecule important properties? Come on.
Water?
Yeah, water has hydrogen bonds. It's important because you wouldn't have liquid water if there wasn't hydrogen bonds. And it is true that if you freeze the water, it's the hydrogen bonds that form the ice. What other molecules have we discussed that have hydrogen bonds that give the molecule important properties?
DNA.
Yeah, DNA. You wouldn't have 2 strands of DNA without the hydrogen bonds. It's true that proteins have their correct folding pattern partly due to hydrogen bonds. And that's true of all the molecules we talked about. We just didn't talk about it much with the other molecules. But the important part is that all of these molecules have important properties because of the hydrogen bonds. And what would happen to these molecules if all the hydrogen bonds were to all disappear? What would happen to 2 strands of DNA? If all the hydrogen bonds would disappear, there would not be 2 strands of DNA. You'd only have a single strand of DNA. And obviously, if all the hydrogen bonds in water were to disappear, we would not have liquid water, we would have steam. All right, any questions about this lesson? If not, that's it. I will see you at 7, no, Yeah, it's actually at 7, isn't it? Let me see if we're supposed to start the next lesson. Today is the 18th. Up, I'm supposed to start chapter 3. So let's begin chapter 3 because I think this class goes to 6:50. Yep. I was thinking we'd stopped at 6:20, but that's a regular term. So let's move on to chapter 3. And I should have talked about this. We're going to talk about chapter 2 and chapter 3, and I don't think I did that today. I will promise we will stop a little early. Let's see. Let me save that. Close it down. Open up chapter 3. All right. Chapter 3 is observing microorganisms. The major goal in the rough outline is shown here. The terms, always know the terms for this chapter. In fact, you should really get to know the objectives, which is more than the major goals. 2, understand the size of organisms and the properties of microscopes. Understand the major difference between a light microscope and an electron microscope. 3, understand why staining improves resolution. And know 3A that there are 3 different types of stains: a simple stain, a differential stain, and a special stain. And then 3B, understand what the Gram stain is, its procedure, and the basis of its differential staining. Any question about the major goals on the rough outline? In chapter 3, we are going to talk about observing microorganisms, and in science, we use the metric system. So our unit of measurement is the metric system. You must know the metric system, and you will be quizzed on the metric system. You can see the end of the study guide for some help on the metric system, as well as this slide here. And let's see if I can show it. I've got something online to help you study or learn the metric system. All right, it's way down on week 3. It's probably because we're only going to start this lesson today. You're going to finish it next week. But anyways, I've got some metric quizzes. When you click on them, this quiz is from outside of the, that opened a little bit, but not very well. It's opening a web page outside of Canvas, and so that will not work in Canvas. What you need to do is click on this link here, and then it'll open it in a separate tab. Let me go ahead and close that down because that's surprising that worked partly. Normally, you see something where it says Firefox can't open this page. And what you do is you just click on this link there, and then you can open it. I'm not familiar with this quiz there, but you probably click on Start here. Let's go to the other one. That quiz has changed a bit. Here's an interesting quiz for you. I don't think I want that. I think I want the Now be careful on what you click on. Like, don't click on the download, click on the start. And then it'll ask questions like the basic unit of the metric system is, oh, excuse me, the basic unit of the metric system for length is meter. Submit my answer. And if it shows green, that is correct. Continue. Oop. I didn't want to click on that. Let's go back. Close that down. Yeah, I don't want to download something on my computer, so be careful on what you click on here. And where the heck is that? There it is. Next. Click on next. Do not click on continue. And then select the basic unit of the metric system for volume. The correct answer is liter, but I'm going to give the wrong answer to show you what it looks like. And it'll show red for wrong, and then liter, and obviously only got 2 out of 15 correct. Click next again. And the basic unit for the metric system for mass is gram. Correct. Next. Metric units have prefixes that represent multiples of the base. What is the prefix for 1,000? That is kilo. Kilo is correct. All right, that's how you work that. Any questions about any of that?
I have a question. Are we using the lockdown browser for this one?
No, you're not using the lockdown browser for this metric quiz because these quiz questions are just for you to practice working with the metric system. Is that clear? So when you hit these links, you will have to click a second link to get it to open. And then be very careful on what you click on because you don't really want to download something incorrect. This one looks a little easier, but don't click on continue. Scroll down. And I don't see where I'm supposed to click. I don't think you want to click there. I don't want to add the extensions to my computer, so that's not right either. Well, I'm not seeing the link here. It may be you need to download something on your computer to get this one to work because I don't see which is the equivalent of 2,000 millimeters. That would be 2 meters. Well, that didn't seem to do anything. All right. Anyways, those quizzes are just to help you. You do need to know. Let me see if I can blow this slide up. You do need to know how to convert from one metric unit to another. So you need to convert from kilometers to meters to centimeters. You don't really need to know a decimeter. In millimeters, in micrometers, and nanometers. Let me see if I can blow this up a little more. Sorry, my eyesight isn't very good. Maybe you guys can read it, but A kilometer is 1,000 meters. A centimeter is 1/100th of a meter. A millimeter is 1/1000th of a meter. So you have 1,000 millimeters in 1 meter. Any questions about that? And you need to convert between all of these. Like I said, you don't need to know a decimeter, but you do need to know a centimeter, a millimeter, because they're on your ruler. The millimeter is the smallest measurement on your ruler. A meter would be on a yardstick, and a kilometer would be something you drive. Let me see. I think a kilometer is 0.6 miles. Is that right? Yeah, about 0.62 miles. And you need to know a micrometer because that's the size we see and measure with under an a regular light microscope. So a micrometer is the size we measure and see under a light microscope. A nanometer is the size you see and what we measure with under an electron microscope. You don't really need to know a picometer. All right. And you do not need to convert from metric units to English. That's hard. You just need to be able to convert between metric units. Whoops, I guess I should have said you don't need to convert between metric and English. That's hard, or US equivalent. You just need to convert from metric to metric. Any question about any of that? For me, I just remember meter is the base unit, 1,000 meters is a kilometer, 1/100th of a meter is a centimeter, 1/1000th of a meter is a millimeter, and 1/1,000,000th of a meter is a micrometer, and then 1/1,000,000th of a meter is a nanometer. And if I need to convert from, let's say micrometers to kilometer, I'll first convert to a meter and then convert meters into kilometers. So I just base everything to meter and then go the other way. You don't have to do it that way, but that's just an easy way to know. And then, like I said, you just need to know the prefixes. Like kilo means 1,000 meters, but kilogram is 1,000 grams. So you just need to know the prefix. And then a meter is your metric unit for length. A gram is the metric unit for weight or mass. And then a liter is the metric unit for volume. Another reason for knowing the metric system is 1 cubic centimeter of water is equal to 1 gram of water. Any question about that? And you don't need to know the other part, and that is if you give 1 British thermal unit of energy to the water, 1 gram of water, you will raise the water 1 degrees Celsius. I guess you should know that water freezes at 0 degrees Celsius and boils at 100 degrees Celsius. So that water is actually kind of defined on the metric system. Any question about any of that? We could talk about the Fahrenheit system. It's actually designed on salt water at 0 degrees Fahrenheit is the temperature that ocean water in Denmark, and I think that was a little bit less salty than regular ocean water, freezes at. And that's why 0 degrees Fahrenheit is less than the temperature water freezes. That whoever set it up, he was in Denmark and set it up on the Denmark ocean water freezing, which has salt in it, and that's why it doesn't freeze at 0 degrees Celsius. I don't know. Nah, I'm not going to talk about that. We're not going to even talk about it. All right, let's move on. The other reason why you should know the metric system is obviously science uses the metric system, medicine uses the metric system. Like when the doctor tells you, take 1 cc of this medication, that's a metric system. And then the third reason we should know the metric system is because this map is showing you all the countries in the world that do not use the metric system. And as you can see, there are only 3 countries in the world that do not use the metric system. And only one of them is a big country, and that's us, the United States. And the United States is actually slowly converting to using the metric system. And one of these other 3 countries is also converting to using the metric system. Any question about any of that? All right. When we're talking about organisms, we say that a eukaryote cell, depending on what the eukaryote cell is, about 10 to 500 micrometers in diameter. A prokaryote cell, this is a large one. Most of them are about 1 to 2 micrometers in diameter. The largest are around 10 micrometers in diameter. And I don't know why they have the largest one here, but they don't have the smallest. The smallest prokaryotic cell is around 0.4 micrometers in size. There are some cells smaller than that, but those are probably non-viable cells. And then a virus, its diameter size is between 0.02 micrometers to 0.44 micrometers. So a virus is about 1/10 the size of your average prokaryote, which is about 1/10 the size of a normal eukaryote cell. Like a blood cell is around 10 micrometers. Don't quote me directly, but it's around 10 micrometers. You'll note that the largest virus is actually about the size of the smallest prokaryotic cell. And then the very largest prokaryotic cell is about the size of the smallest eukaryote cell. The largest virus we're going to talk about is megavirus and Ebola. And I should mention that Ebola is the longest virus, which is about 1 micrometer long. But Ebola is a very long and skinny virus, so it's not the largest virus, at least in the diameter. The largest virus in the diameter is megavirus, and it's about 0.4 micrometers in diameter. Any question about any of that? All right, I've already talked about this. You should know the rank order of things, the resolution of what we can see. Blow this up. So humans, about the biggest thing we're going to talk about. It's bigger than a chicken egg. Remember, the yolk of a chicken egg is the largest cell that most, if not all of you, have ever seen. And for me, it's also the largest cell I've ever seen. I've never seen a turkey yolk, and I've certainly never seen an ostrich yolk. Frog eggs is about the smallest thing we can see with our naked eyes. Below the size of a frog egg, you really need to have a light microscope, which is a eukaryote cell size, and then a bacteria size. Smaller than that is the virus. Smaller than a virus is a large molecule like a protein. Smaller than a large molecule like a protein is a small molecule like water. Smaller than a small molecule is the atom. Any question about that? And then note, if this chart has a mistake in it, that's one micrometer there. It's actually 1 millimeter because 1 micrometer is here. All right. Any questions on any of that? I think I'm going to stop here and we'll talk about the microscope next time. Any questions? All right, if there's no questions, I'll see you at 7 o'clock for the lab. Come on. Ah. We're just going to leave it. I like to make that bigger, but we're not going to be able to do it easily, so. All right, I'll see you in the lab. Assuming I can get this open.